send you the slides when I'm done. It's not a problem. I didn't mail them through because the deck is quite big, but I'm happy to share them afterwards. No hassle. So you'll have what you need, I'm sure.
Great. Thank you very much.
Okay, cool. All right, so where are we in terms of malaria? There's a picture of malaria back as what it looked like in 2006. In terms of the actual infectious areas, not much has changed, in fact. So back then, we were looking at about half the global population at risk, 243 million new infections and about 1 million deaths. And where we are now is better, but not necessarily better. So 2020, we recorded around 240 million new infections, so about the same, but only just over 500,000 deaths. Now, it's still 500,000 too many, bearing in mind that also, of course, most of the victims of malaria are kids younger than 5. But certainly, we've come a long way. I can tell you when I did my honors project back in 1997, we were looking at 2 million deaths a year. So we're down to about a quarter of that in the last 25 years. So we're moving in the right direction, but probably not necessarily moving fast enough. But anyway, we do what we can. This is what the malaria life cycle looks like. As you can see, it is super complicated. We've got 3 major stages. There's the human blood stage, the asexual blood stage, which is pretty much where all the malaria symptoms are and where all of our current antimalarials find themselves being aimed. In addition to that, there's another blood stage, which is the first round of the transmission stage. The parasite is mutated into has generated a different form of itself, which is now reinfectious to mosquitoes. That can then be picked up by the mosquito, where there is a further fairly complicated life cycle, which goes through the mosquito's gut, ends up in the salivary glands. Parasites are then injected into the next person who gets bitten during a blood meal. Parasites invade the liver, and 8 days later reinfect the blood, and we start our cycle all over again. I'm not sure how much you guys have seen under a microscope. Up here on the top right-hand side are pictures of red blood cells. So basically, a normal healthy red blood cell looks like a little donut, sort of concave on both sides and kind of flat. Once it gets infected by a parasite, it starts to swell up a bit. So you can see here this middle picture on the right-hand side. The red blood cell starts to swell. What's happening at that stage is the parasite is basically chewing out all the hemoglobin and the internal cytoskeleton of the cell to make space for itself and to use that as a nutrient reservoir, a source of food, basically. And by the time the red blood cell is actually ready to burst, the parasite has gone from one little ring, which you can see here, into this fat trophozoite, which is what's starting to form under here. And then it divides into a whole lot of little merozoites in what we call the Schizon stage. And depending on which strain of the parasite you've got there, it may be 8 merozoites, it may be as many as 30 merozoites. And at this stage, the parasite is big and bloated. It looks like a beach ball. The structure is really weak. At some point, it will burst, and all those merozoites are released into the bloodstream, and this cycle will continue. It is at this point that you get all the clinical symptoms of malaria, the 2-day fever, the general illness. When you have the liver stage or the gametocyte stage, you're completely asymptomatic and will go about your day with absolutely no issues whatsoever. You won't even know you're sick. Okay, so that's where we are currently, just as a little bit of a background. The bulk of the work we're going to be talking about today carries on here in this human blood stage. When you guys send compounds to get an IC50, that's what we're looking at. There are other groups who can do the liver stages. Your collaborator up in Pretoria, Lynn-Marie Burkholz, her group works almost exclusively with gametocytes, and she's one of the best in the world at that. So she probably would be worth getting her in for a webinar at some stage, because we'll touch on gametocytes today, but they're really, it's a huge amount of stuff you can do with that they are very good at up there. I mean, there are groups who do all the mosquito stage work as well as the liver stage. But here at H3D, we're primarily involved with just the asexual blood stage in this erythrocytic cycle here in the middle, although we will talk through the whole lot. Okay, in terms of drug discovery, again, there are many different ways you can skin this particular cat. I'm going to be looking at the way we do things here at H3D as a sort of the context for this talk in general. Yeah, jump in.
Yeah, so does that include the gametocytes assays as well?
Oh, we don't. Lynn-Marie does, but we will talk about the gametocyte assays in this talk, yes.
Oh, okay. All right.
Okay. So we'll talk about all the assays. We'll talk about the liver stage. We'll talk about all the mosquito stages as well. But in terms of the assay work, H3D doesn't do gametocyte work. We just do the other stage. Okay, cool. So if you are involved in drug discovery and development, basically, this is the way we handle things here. We start with target validation. In our case, our target is the malaria parasite. We have the what we call the phenotypic screen, so the whole parasite growing in red blood cells. We also have a mouse model here for in vivo work. We know that causes malaria. It's a fully validated target, so that's easy enough. We will then get into compound screening, either from a library or somebody will bring us some chemical matter to work with. We'll go through a raft of sort of circular assays where we generate data, feed it back to our chemists who will design the next round of compounds as an iterative process. At some point, good compounds will go into secondary assays, a little bit more advanced. For better compounds, hopefully at some point we'll have something that's good enough to go into an animal model, either a healthy animal or a disease model in order to generate PK and proof of concept data. And then hopefully at some point we end up with a candidate. All of that falls under the drug discovery part of drug discovery and development. Once you get any sort of human trials involved, that moves into the development start of drug discovery and development. And that is nothing we do at H3D. That's always passed on to an industry partner. It's also at that stage where things become really, really, really expensive. So good to know that it's someone else's money that's being spent at that point. Okay.
Bill.
Yeah, jump in.
Please say a line about the ex vivo. On your previous slide, you have an ex vivo assay, a secondary assay. So.
Okay, so there are times where you might want to work with something that comes out of a live model and put it into an in vitro model. What we've done previously is we've got mice that are infected with malaria. You would then dose the mouse with the drug or whatever you're trying to do, and you would take samples from that and then work with those samples in an in vitro context. So they've come from a live system as opposed to a cultured in vitro laboratory setting, is what we refer to as ex vivo. So out of a live model into an in vitro system, but as part of the same experiment.
Thank you.
Right. That makes sense. Okay, cool. All right. The important thing to note is that we don't work in a silo, right? The malaria community is quite big. For many, many years, the malaria community was a bunch of people who were doing their own thing. Some years ago, MMV, Medicines for Malaria, entered the scene, and their big goal was to try and produce drugs as opposed to just doing malaria biology. And they really have worked tirelessly over the last 20 years or so to bring the community together and to try and set a set of guidelines and standards. So we're all really, we thought we were pulling in the same direction, but you realize actually maybe you need someone to drive it a little bit and really just try to focus the efforts. So currently MMV have what they call TCPs, target candidate profiles, and target product profiles. And these basically are guidelines as to what they think each drug should be doing. And this is where their funding goes. So they're interested in finding compounds which we would call TCP1. These are compounds that clear symptomatic infections, all the blood stage parasites, something that when somebody comes into the clinic sick, you give them a drug, it cures them. They're also interested in TCP3. So for certain types of malaria, particularly Plasmodium vivax, that leaves remnants of dormant malaria in your liver, which can actually reactivate months or years later. And they're trying to find a way to kill that. But none of our drugs work in that area. They're looking for drugs that can work prophylactically. So basically, when the infection gets itself established after the mosquito bite, something that can kill it there already before you even get sick. They're looking for molecules that can block transmission, either at the gametocyte stage in the blood, or even once it gets to the mosquito stage. That's a bit more complicated. We'll talk about that later on. Beforehand, nobody was really looking at things like this. And MMV have sort of said to us, look, these are the areas we're interested in looking at. If you've got compounds that you think can do this, let's put something together and try and get this compound to the drug stage, get it into patients and see what we can do with it. In addition to that, we know what each of the molecules would do. In the modern era, nobody uses a single drug anymore. If you look at something like tuberculosis or HIV, it's always a combination treatment. And that's important to get around things like drug resistance. So what MMV are hoping is we'll produce a couple of different products. Target product profile 1 is for case management. Somebody comes in sick, you give them a tablet which contains 2 or more drugs. Hopefully with some TCP1 activity in there, maybe some TCP5 or TCP4 or 3, depending on what sort of area you're in or what sort of work you're trying to do. They're also looking for something which is for protection purposes. What they really would like in this area is something you can give somebody maybe by injection that will give them coverage for 3 months. So somebody who's going, at the start of the malaria season, we could, dose a whole lot of people with a drug that keeps them safe for the entire season instead of having to take tablets once or twice a week to keep the malaria at bay. It's worth noting at this point that to this date, pretty much every single antimalarial on the market for the last 100 years has all worked in TCP1. Beforehand, we weren't doing any work at all in the other areas. This is really something that MMV and through their partners, Melda, the Gates Foundation, really trying to get the community behind, thinking outside of the box and trying to get something that works, bigger picture, than just curing sick people. we'll talk a bit about that going further as well. Okay.
So Dale,
yeah, jump in.
So in a drug discovery project, and you find an active compound, do you then try combining that with another existing drug immediately, or you want to find as potent as possible?
Yeah, we worry about all that right at the end. So when it looks like you might get towards human trials, someone will start thinking about, right, which would a good combination partner be for this drug? But that's something we worry about much later.
Right.
Okay, cool. Okay, so as Siobhan mentioned, we've been in the game here for a long time. We put our first malaria screening cascade together back in 2010. This is what the cascade looked like in 2017 for us. You can see there's a whole lot of different assays being done. If you look at the key down on the left-hand side, it shows you which department each assay is done in by the coloring on the outside of the block. It also shows you anything where it's written in black, something that we do in-house. Anything where it's written in green is something that we outsource to one of the partners. There's no point in every lab being able to do 50 different assays. It makes much more sense for each lab to pick up 4 or 5 good ones and leave the other ones to the experts who can really specialize in that. So we've never tried to sort of get to the point where we can do every single assay on this page. You need a department that was 500 people deep to get that right, and it's just not really worth the effort. Rather, we stick to the stuff we're good at, and we rely on guys who are good at other things to handle that for us, and that's really a good way to work. Something for you guys to think about, at the Ghana Drug Hub as you guys go forward. So that's what it looked like back in 2017, and our aim here was to develop an orally active compound for blood stage parasite clearance, TCP102. You'll notice on the previous slide there was no TCP2. That's been actually phased out completely as something which was a going concern. TCP2 compounds were compounds which hung around for a long time and acted slowly. We've now decided that's not necessarily of value to us. Everything has got to be fast acting and hang around for a long time. So TCP1 and 2 have merged into the new TCP1. So TCP2 no longer a concern. So that was the story in 2017. Where are we now in 2024? Well, that's our current cascade. And you can see it does look kind of different. So the colors are different. We've got away with some assays. There are some assays on here like this one, which are brand new. They didn't even exist back in 2017 when you put the other cascade together. This tool here, MMV Solar, wasn't a thing. The NSG model, not really as widely used as it is down here at the bottom. So things have changed a lot. And in fact, our current story, our current aim now is to develop an orally active compound for blood stage parasite clearance, TCP1, which also has an exceptionally long half-life and also has an exceptionally low resistance risk, and ideally has multi-stage activity covering TCP 3, 4, 5, or 6. So really that's where the bar has moved to. Back in 2017, a drug that sort of works, hey, it's happy days. Now these are the sort of things that will be regarded as a minimum requirement for new drugs. It's also worth, as I mentioned on the previous one with our TPP2, they are currently, for the first time in a long time, thinking about what could we make do with a long-acting injectable as opposed to a drug. We know what the environment is where malaria tends to find itself. It's often out in the rural areas. We've got to have compounds which are stable in tablet form that can survive at room temperature. It's nothing that needs to be administered via a nurse with a needle, nothing that needs to be stored in a freezer, because sometimes, some villages, they just don't have those facilities available. This is the first time actually that people are beginning to realize that maybe we need to find a way to make that a reality. So for the first time in a very, very long time, we are considering something that isn't a tablet and actually, will be injected, provided it can give 3 or 6 months worth of clearance. So this is a sort of a pilot project, which is new for MMV and all the partners, but it's an exciting area to be in. Okay, cool. So we'll talk a bit about some of these assays going forward. I'm going to focus primarily on the biology ones, which are these ones here, and this guy down at the bottom. The other ones are all ADME-based or chemistry, which is a little bit more your guys' area than mine, but we'll talk about that as we go along. What is important to note is that we put our assays into a couple of different categories. So we talk about tier 1 assays, right? Primary assays. These are used primarily for quick decision-making, basic SAR. Pretty much every compound you make will go through tier 1. It's the rate determining step of your process. You don't do anything without that result for the most part. And for the most part, this will have your primary screen against your pathogen there. Yeah, jump in.
Yeah, so on the previous slide, you did say that MMV is now looking for long-acting injectable also as desirables.
Yes.
This kind of new information to me. I thought all the TCPs were more of oral tablets?
So it's basically, they're looking here for this TPP2 will probably be a long-acting injectable. So what they are hoping to try and do at some point is get to the stage where at the start of malaria season, you can give people something that will last for 3 or 4 months, as opposed to worrying about taking a tablet. It's not their primary aim, but they're really trying to put a lot of their resources into getting something that can do this, as well as the TCP1 stuff.
Okay.
Okay, like 5 years ago, nobody would have even considered this. Is how new it is.
Yeah.
So this is in their newly published sort of roadmap for 2024 to 2020 to 2030, which they published in June. So it's hot off the press, brand new for all of us.
Okay.
Okay. Cool. All right. So tier 1, as mentioned before, this is probably going to include the primary screen against your pathogen, right? There's no point in making compounds and sending them into all sorts of dozens of other assays if you don't know if they can kill the malaria parasite. So generally, your tier 1 assays will always have your primary screen in them, which is against the pathogen, whether that's in vitro or in vivo or whatever the case may be. It all depends on your disease area. In our case, this is the whole cell screen against NF54 to get an IC50. Okay, moving on from that, there are secondary assays, which we call tier 2. Fewer compounds go into these assays, but they generally are all on what they refer to as the critical path. Okay, so they're all essential assays. It gives us a much deeper understanding of SAR, and will typically include much more advanced DMPK and occasionally mechanism of action assays. When you find something that actually just is an antimalarial, we don't worry too much about trying to find out how it works. Does it work? Yes or no? That's the answer. We worry about the mechanism of action and trying to find out how it works properly much later on down the line. So that would be a sort of an example of tier 2 type assay. Then we get into the tier 3 assays, and these are only for very, very advanced compounds. In the olden days, we wouldn't touch a tier 3 assay at the start. These days, they'll carry out a couple of these tier 3 assays right at the beginning of a program to get an idea of how good a series may be. So they'll do it right at the beginning, try and work out, right, do we have liver stage or gametocyte activity? Do we have a resistance risk, yes or no? It'll then not be looked at again until significantly further down the process. So your really, really advanced compounds, probably 2 or 3 years, maybe even further into your program, you'll start calling for tier 3 type assays. Particularly also quite expensive and complicated assays like the CACO assay, like the NSG mouse model, which is basically a malaria disease model. You know, if you're producing 400 compounds, that assay takes 2 months to do. You're not going to put all 400 of your analogs into that. You'll be sitting here till you're all dead of old age and you won't have the data back yet. So all of these are only for really, really select advanced compounds that tick many, 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 many of your blocks before you worry about things like that. Tier 3 may also include some of the critical showstopper assays, things like the genotox test or the Ames test. It's at this point, you'll look at field isolates and resistance panels. You'll also need to figure out whether you have a HERG issue. HERG is a complicated one. It's basically a cardiac channel that controls the beating of your heart. And essentially, you don't want to be giving someone a tablet that can send them into an arrhythmia, which may cause the heart to stop. So generally, a bad HERG signal, if you pick it up early, you'll push that from a tier 3 assay probably into a tier 2 assay, because that can kill your program stone dead if you cannot solve your HERG issue. Okay, cool. All right, so now we've sort of set the scene. How do we actually do these assays? What are we looking at? Yeah. So it's important to note that we only, believe it or not, even though we've known about malaria for hundreds and hundreds of years, we've only been able to culture the parasite since 1976. Before that, all malaria work was done in animal models. They did it. You would be surprised at how much malaria work was done in chickens and ducks. You'd be surprised at how much malaria work was done in mouse and monkeys. And in fact, in the olden days, of course, we didn't even bother to test, right? If something worked, it went onto the market. We didn't do any sort of safety testing, nothing. So something like chloroquine probably had very, very little testing done on it at all before it was rolled out as a mass market drug in 1931. Nevertheless, since 1976, we have been able to culture the parasite. And that's sort of generally a useful thing to do, because I promise you, it's way, way easier to do work in a test tube than it is to work with, 100 chickens or ducks or mice. So this is where we find ourselves currently. What you can see here on the screen is an example of what a malaria culture would look like. So we talk about the ring stage. You can see that on top there, the parasite looks like a little diamond ring. There are lots of rings in this slide. There's also the trough and Schizon stage, which is these 4 dark, dark purple big things that you can see here. Generally, this is a process which takes about 48 hours to happen. So you start with a ring. After 24 hours or so, you have a small trough, which becomes a big trough, which then bursts. And after 48 hours, you're back to the ring stage. So that's an idea of what we would find in the lab. Generally speaking, we use a couple of major parasites. So these are our three as our primary and secondary parasites. NF54 is the general wild type, isolated from in Amsterdam, of all places, would you believe? Then there are two resistant strains, DD2 and K1. The origins are listed there. It's a little bit more complicated, but that's fine. We have access to a battery of other strains, which you can see listed up top here on the right-hand side. So there are some generally drug-sensitive ones. That's D10, HP3, FC27, 3D7, RSA3. The numbers aren't important. It's just things you may find in the literature. And then there are some quite resistant parasites under there as well, W2, FAC8, RS11, and 7G8. There are some important differences between these, but again, often, and this is really a little bit more relevant in the sort of academic biology context more than the drug discovery context. So don't worry too much about it. Generally speaking, you'll want to run your compound of interest against the wild-type parasite, one of the multi-drug resistant parasites, and then later on, it'll be put against a sort of a current field resistance isolates panel to see if you get an idea of what the drug might do out in the wild. Okay. What I would like to draw your attention to is this paragraph down at the bottom here. Just for the benefit of your biologists who probably aren't sitting in on the call, these guys, if they are doing a lot of work for you, probably are coming into the lab 6 days a week, sometimes 7 days a week. The assay works 9 times out of 10, but you will have times where things just don't work and it's generally a bit, and the data that comes out of the lab doesn't look promising. I'm sure you on chemistry, sometimes work tries, you can't get something pure, something doesn't crystallize properly. It's certainly not a unique thing. We all have bad days when it comes to the lab, yeah? So please, give your biologists, keep them at the back of your mind when you're sending them 100 compounds to test as to exactly how much work they're going to be doing to get it all done. Generally speaking, we would maintain the cultures at around a 3% hematocrit. The hematocrit is the amount of red blood cells in the tube relative to the amount of growth media in the tube. So 3% volume by volume, 3 mils of blood per 100 mils of growth media, and we'll be looking at maximum 10% of those red blood cells will be parasitized. The reason we can keep it only that low is because otherwise the parasites burn through the nutrients too fast, and you need to culture them multiple times per day to keep the culture running optimally. In order to keep it that low, we do have to dilute pretty much every second day with fresh red blood cells. So on a day when your culture is primarily these big fat troughs, so I lost my cursor, these big fat guys that are about to reinfect a whole lot more cells, we'll give the culture a 5 or 10-fold dilution with fresh blood. So pull out 300 microliters of those 3 mils, chuck in 2.5 mils of fresh blood, and there you go. That'd be the culture good for the next sort of 2 days or so for the assays that you're going to use it for. This diagram down at the bottom here is from a mouse, in fact. And what you've got here are red blood cells. These are the gray ones with parasites inside them. These two big guys you can see here are actually white blood cells. So I'm not 100% sure if I've got this right. I think this one here is a basophil, and I think this one here is an eosinophil. We don't tend to keep those in our parasites because we work with cultured washed red blood cells. There's no white cell component whatsoever. So we will very, very, very infrequently, maybe once a year, see one of these cells in our cultures, and you generally don't find them. If you're working with mice or monkeys, absolutely, you'll see these sort of things all the time because obviously you're taking whole blood from the animal, which is full of white blood cells. But ours don't, no white blood cells at all in our cultures for the assays. Okay, the cultures obviously have to be kept sterile. Yeah, so
Neil.
Yeah, jump in.
Yeah, so when we send compounds to your lab, we get NF54.
Yes.
Right? When we do the assays locally, we get 3D7.
Okay.
How do they compare? And also in terms of even different labs, and what do you advise?
Okay, so we've done some collaborative work and some comparative work with your lab. For the control compounds, we get about the same IC50s. So, don't worry too much about the values that you're getting out of there. Your guys up top, they know exactly what they're doing, and technically the assay is fine. In terms of the parasite, there will be some slight differences. So NF54 was isolated from an airport worker in Amsterdam. He had never traveled, so they think it was a mozzie that came on a plane from Indonesia that bit him coming out of the plane and then died because it wouldn't have survived the climate in Holland. Some years later, they isolated parasites from NF54, and that was called 3D7. So the two parasite lines are very, very, very closely related. If you like, you can sort of think of 3D7 as a great-grandchild of NF54. But in terms of the drug profile, they are almost identical. So 3D7 or NF54, they're almost interchangeable. You'll see some slight differences between compounds, but nothing that you need to worry about in terms of the data. Everything that you see in NF54, you can assume will be probably within 5 or 10% the same in 3D7. Nothing to concern yourself with there.
Thank you.
Okay, cool.
Hello there.
All right. Yeah, jump in.
Hello. Yeah, so same on this on that slide about the clones and the strains.
Yes.
Like how do you differentiate between them, and what constitutes a wild type 1 and a wild type 2?
Okay, we're going to come to that on the slide after this next one.
Okay.
Okay, cool. All right. So yeah, so this is what a laminar flow cabinet looks like. Essentially, it's like a fume hood, but the air blows outwards instead of inwards. The idea here is to protect the culture, and actually, because you can't get malaria by breathing it in, we don't have to worry about protecting the operator. So essentially, you can see up top here, dirty air from the room comes in, it gets filtered and comes out sterile to keep your workbench sterile. That helps keep the culture sterile. We don't want obviously any bacteria or any fungus or anything like that getting into the culture, because that will skew your results completely, as well as killing your parasites eventually and killing all the red blood cells. So it's got to be kept super, super, super sterile. The basis of our growth media is something called RPMI 1640, and it's basically like Powerade or Energade, with some salts and sugars chucked in. It's a sugar solution with some other minerals and some amino acids, and generally designed to keep mammalian cells of any sort growing quite happily. It's also designed, the pH inside there is designed to be kept stable by addition of bicarbonate afterwards. This is quite important because obviously, as you know, you'll have plenty of carbon dioxide in your blood, which forms small amounts of bicarbonate. So the parasite is used to regulating its own pH by shifting protons and bicarbonate around inside itself. So we use bicarbonate as well to buffer the media here. You get 7 different types of RPMI. That's not super important. The biologist will find that more important. You can add different additives to it. Not the end of the world. The important thing for us to note is, really the most important thing, is we look at the color of it. It's got methyl red in it as an indicator. So that's got a fairly narrow pH window. It's got to be used at around pH 7.4. If your pH goes above 8 and the parasites are not growing absolutely brilliantly, you'll kill your culture. If it goes below, probably below 7, you'll kill your culture again if the parasites aren't growing well. It's too acidic or too basic for them to regulate. So as long as the medium is orange, your parasite should be growing fairly happily, ticking along quite nicely. These are the other things you need to add to it to make it grow. Again, this is here purely for interest. Don't worry about exactly what's in there. Important thing to note is it's got some white blood cell components, just that there's some lipids in there for the parasites to work with. It's got hyperxanthine, which is a DNA precursor. They would normally scavenge that from the host's blood supply. And obviously, we don't have any of that circulating inside of our donated red blood cells from the blood bank. So we've got to add that manually, otherwise the parasites can't proliferate. Okay, so good question was asked previously, which is, what is the difference between a clone and a strain? All right, essentially, every time we isolate parasites, we start with a strain. A strain is a mixed population of parasites, and a clone is a genetically identical population of parasites. How we do this is an interesting story. So we start with our patient, who, as you can see there, is developing malaria. You see all those little purple spots forming. Someone comes to us and says, look, we think this guy's got malaria. Do you want to take the parasite and culture it? We'll say, yes, please. So, sorry, my system is frozen here. There we go. All right, so we draw a small amount of blood. We add some medium. We add a significant chunk of time, and we hopefully end up with some parasites growing in a test tube. And if you leave that for a couple of weeks, hopefully you end up with a few more parasites, and then hopefully lots and lots of parasites. At some point, you'll want to characterize these and find out, right, what do we have here? Is it wild type? Is it generally sensitive to all the drugs? Is it resistant to a few drugs? If yes, which ones? At this point, you may try to genotype and find that you've got a couple of different genetic variants in there. It's a bit of a mix and match, a bit of a bastard population. So what you'll then do is go in with a micropipette and pull out one single parasite. This is all done under a microscope. Again, we'll add some medium, we'll add another long chunk of time, and then hopefully we will end up with a genetically homogeneous population. And that is what we refer to as a clone. So in the strain, you may have a couple of different variants of small things in here that may or may not affect the way the parasite grows. With a clone, you should have a pretty much a genetically pure, identical population. Okay, I hope that makes sense to everybody. Again, in terms of the big picture, it's not super, super important. What's more important is that you know what your strain is doing. Is it sensitive to your drug, yes or no? Is it sensitive to chloroquine, yes or no? Is it sensitive to any of the other compounds in development, yes or no? That'll give us a general idea as to whether or not the strain is suitable for the sort of work that we're doing. So whether it's a strain or a clone actually is neither here nor there. Sorry, give me 1 second. I just got a call coming in. Hello? Lee? Lee? Okay, I think I just got pocket dialed. Never mind, not important. Okay, sorry about that. Okay, cool. All right, so that's kind of what we're doing in terms of getting the parasites to grow. How do we actually get the... Yeah, jump in.
Yeah.
Yes.
So please, on the previous slide. So do you have to do this every time you get new blood samples, or?
No. So this is not something that we do, but this is how we got all those strains that exist, sort of that we use, all those ones I mentioned on the previous page. And these are how they were all brought in. If somebody has malaria and you wanted to get it and try and genotype it, this is what you would do. But no, we buy our reference strains in from the malaria repository. So we say, right, send us a vial of NF54, send us a vial of K1, send us a vial of DD2, and then we'll grow that for the year. And that becomes our stockpile. Yeah, okay. So if you want to isolate parasites from a patient in Ghana, this is the process you would go around. And this was done for our ones before we got hold of them years ago. Okay, cool. Okay, so essentially, what are we trying to do with this assay? So a basic principle of all anti-organism assays, whether you're trying to do general antibiotics or tuberculosis or malaria, whatever the case is, you're going to get a hold of your organism in culture. You're going to expose to various levels of the compound for a specific period of time. In our case, that's 72 hours. You'll determine the level of survival, and from there, calculate your IC50. And the IC50 is the concentration at which half of your culture has been killed. And you can use this to rank your compounds. So we've got 20 different compounds. We've now got 20 different IC50s. And we can say, all right, well, which is the lowest one? Which is our best performing compound? And that's what we're looking for. This assay was done originally using radioactive hyperxanthine as a measure. As mentioned on the previous slide, we've got to add hyperxanthine because the parasite can't scavenge it from the host. It doesn't have a host if it's growing in vitro. And this is a DNA precursor. So as the parasite grows, it incorporates this radio-labeled DNA, and then you count how much radioactivity is left. And that will give you an idea of how many parasites have survived. Okay, this is how we did things back in the day. Of course, it's difficult to work with radioactivity in the modern era. It's difficult to dispose of, store it, even if it's only tritium. So things have moved along. We now use newer assays such as CyberGreen, which is the one you guys use up north at Noguchi and UG. What this does, it's a dye that basically binds to DNA. If you've got parasites, they glow in the dark, which is hell of a nice. Means you can basically, check your culture under a fluorescent lamp, under a fluorescent laser rather, and you look for anything that flashes green at you, and you know, right, there are parasites in there. You can count how many have survived. And that's how you guys generate your IC50s. We do ours with a dye called nitroblue tetrazolium using parasite lactate dehydrogenase. And ours basically measures metabolic activity of live cells. So we chuck some stuff in there. If there are living parasites, they're able to basically make the dye change color. And we're looking for the color change as our marker of surviving parasites. Okay, don't stress too much about it. This is just the biochemistry of the assay. So there is something of our assay, this is the LDH assay, something called the glycolysis pathway. If you've done any biology in undergrad, you'll know about glycolysis. Essentially, you start with glucose. It goes through a number of different steps. And at the end of the day, you produce pyruvate and 4 molecules of ATP. This is why they tell you, if you need energy, you've got to take some sugar on board, right? You put in the sugar, and bam, you end up with some metabolites and some energy in the form of ATP. So that's all well and good. Why is this important? Well, basically, the parasite produces huge amounts of pyruvate. If it overproduces pyruvate, it back converts pyruvate to lactic acid. And this is an enzyme that works in 2 different directions. If you give it pyruvate, it produces lactate. If you give it lactate, it produces pyruvate. So because this is done by surviving parasites, if we load the parasite full of lactate, it will convert all that lactate to pyruvate. In doing so, there's a cofactor called NAD, nicotinamide adenine dinucleotide. It's a very common sort of cofactor used in many enzymes in biology. It reduces that to NADH. If we put our yellow dye, this tetrazolium, in the presence of NADH, it oxidizes the NADH back to NAD, and it's reduced to a blue salt called formazan. So essentially, if there are live parasites, we give them some lactate and we give them this yellow dye, they will produce purple dye, and we can see where all of our parasites are. And that's the principle of our assay, to try and work out, right, do we have survival, yes or no, to calculate an IC50. Okay, so this is what a typical assay plate would look like.
Dale,
yeah, jump in.
Please, will you please go over the assay again, please?
Okay, tell you what, let me do it from here, and we'll come back to the biochemistry if it's important. I think visually is going to be a little bit easier than worrying about the actual chemistry behind it. Okay. All right, so we're going to start here. Essentially what we've got here in column 1, we've got red blood cells. As you can see up top there, red blood cells, no parasites, no drug. So that tells us what no parasites looks like. It essentially shows us what the assay would look like if everything was dead. Yeah, 0% survival, no parasites. In the second column, we have parasites with no drug. So that shows us what the assay should look like if everything survives, 100% survival. So we now have a 0% survival, we have a 100% survival. What we're then going to add to this plate are 3 different sets of drugs. We're going to start with a high concentration here and dilute them the whole way down, 2-fold dilutions. All right, so we should end up with basically a 500-fold dilution range. So if we start at 6 micromolar, our lowest concentration will be 12 nanomolar. If we start at 2 micromolar, it's 4 nanomolar. If we start at 500 nanomolar, it's 1 nanomolar. It gives you a good range to get an idea of where your drugs are active. We leave that in the incubator for 2 days. We chuck in the lactate, we chuck in the dye, and we wait to see what happens. What hopefully happens is we get that. Okay, so now we can see here where there were no parasites and no drug, we've got 0% survival. So again, my system is stopped. There we go. We can see we get this pale pink color here. So that's what no survival looks like. Here we have parasites with no drug. That's 100% survival. So we've got 2 different shades of purple there. And then from highest concentration to lowest concentration, we're looking for where the color changes from this pink to this purple. And that's basically where our IC50 is. Okay, so you can look at it on the plate, which is all well and good. And that's where our 3 different IC50s are for our 3 drugs. So for drug 1, in the top 3 columns, our IC50 color change is around column 4. For the control drug here in the middle 2 columns, it's here around column 6. And for drug 3 in the bottom 3 rows, our color change is here around position 7. Okay, so we can now visually see exactly where those IC50s are, but of course visually is not very accurate. So we'll then put this onto a spectrophotometer to get an IC50 result, or rather get an optical density result, and from there generate a survival curve, which you can see on the right-hand side, and fit a nonlinear regression to it, and from there actually calculate an actual IC50 value. And that's how we generate your IC50s. You guys would do much the same, but with cyber green, so you won't see it visually like this. It can only be picked up on a fluorescence reader. So the benefit of our assay is that we can actually visualize before we get the result. We can see where it should be, and then correlate that with the graph afterwards. Okay, does that all sort of make reasonable sense to everybody before we go back to the biochem? I hope so. Assuming yes.
Yes.
Cool. Okay, so essentially what we're going to do is would be the normal glycolysis pathway carried out by living parasites. You give them sugar, they produce this pyruvate. If there's too much pyruvate which they produce, they back convert it to lactate. So what we would do is we load the system with lactate, and the parasite will then convert that forwards to pyruvate. Okay, that sort of makes sense to everyone so far. So when we need to see if we've got surviving parasites, we will chuck in a mixture of lactates and this yellow dye. Let the parasites convert the lactate to pyruvate. During that same process, the cofactor that's required by this enzyme is NAD. It's converted to NADH, so it's reduced. In the presence of reduced NADH, the NADH can break down this dye from the yellow dye and convert it to this purple salt. And this process cycles. So lactate produces pyruvate, produces NADH. NADH breaks down tetrazolium to formazan, reproduces the NAD, which is then used with the lactates to produce pyruvate. So this carries on, and you go from a yellow dye where there are no parasites to, if there are lots of parasites, you should end up with purple salt. And we can then hopefully see that here. So the yellow dye, essentially in the presence of the red blood cells, ends up being slightly more orange. That's why it's not yellow here. It's sort of a pinky orange. And of course, the purple dye, well, you can see there the purple salt, it's nice and purple. Does that make sense to everyone? Okay, I'm going to assume that's okay. Brilliant. Okay, so that's how we generate the IC50. And as I said, you can do this with the radio-labeled hyperxanthine method if you want to. If you've got a beta counter, and you're happy to work with radioactivity, you do exactly the same thing, but you'll be putting in scintillation count values into this table here. Same with cyber green, it calculates fluorescence counts, and at the end of the day, it's always converted into this graph where we can read off an IC50. Okay, that's all well and good. What happens if you've got a library of 20,000 compounds? And you don't want to have to run 20,000 compounds through your IC50 assay, because I promise you, you'll be there till the cows come home and then some. So what you can do is use this exact same assay for single point experiments. So you could put 80 different compounds onto your plates, one of each, run them at 2 different concentrations in 2 different plates, add the dye, and you'll get a heat map like this. So what we've got here, instead of 3 compounds that we then serially diluted across our plate from left to right, we've got 80 individual compounds, each one at 20 micromolar. We've added parasites, we've incubated it for 3 days, we chucked in the dye, and from here we can see, all right, well, anywhere where it's this sort of pale pinky orange, everything is dead. Everything where it's this dark purple, everything has survived. So that tells us, right, of these 80 compounds, pretty much anything that's pale, all these ones here, are where you have some activity. And that's how you would do a high throughput screen of a large library of 200,000 compounds. Try and do them 80 or 320 at a time to really try and push those numbers through, as opposed to 1 at a time by IC50. Okay, so you will then convert this plate to a heat map. You decided what your gates are. So the map at the bottom here, everything which is green is where we've got greater than 50% survival at 2 micromolar. So we're interested in anything with less than 50% survival at 2 micromolar. These are all the ones that show up in orange or yellow or red. And we would then know, right, okay, put those results together. You can rank them in Excel, which is very, very nice. Of our 200,000 compounds, these are the 300 most active. And that's the sort of way you would do that as a single point screen. Exactly the same assay, just doing it slightly differently to generate a single point kill, how much inhibition you get at X concentration, as opposed to generating a full IC50. Okay, I hope that makes sense to everyone. All right, so that's how we deal with basic parasite IC50. What about cytotox? Okay, because you always do selectivity, right? So these are done with a variety of different mammalian cells. They're cultured less frequently than parasites. They grow slightly differently, different choice of media, no problem. Essentially, you start with a whole lot of floating cells. You put them into a flask. They should eventually attach and elongate. And at that stage, we refer to our cultures being confluent. When they are confluent, you can take them out and you know that they're growing well enough for you to do your assay. And we do pretty much exactly the same type of assay as we did with the parasites, right? Seed a plate with the cells, incubate with drug for 2 days, chuck in some dye, wait for a color change. Slightly different in this regard. The enzymes required are a little bit more complicated. So this takes about 4 hours for the color change to happen. For our parasites, it's only about 15 or 20 minutes. But the principle is much the same. Obviously, for our parasite assay, we want a very, very low IC50, right? You want to know that you don't need much drug to kill your parasite, 1 or 2 nanomolar, happy days. When it comes to cytotox, you want a really, really high IC50, right? Because you don't want to be killing parasites and cells at 1 nanomolar. You want to be killing parasites at 1 nanomolar, but that same drug must be completely inactive to 100 micromolar against the cells. So you've got a good wide selectivity window. All right, that's what it would look like. Essentially, when we've added our drug and we've given it 2 days of incubation and chucked in the dye and waited 4 hours. So much the same sort of thing. We've got no cells, no drug at the bottom here to show us what 0% looks like. Row G here is cells with no drugs. That's 100% survival. And then we've got 4 different drugs. Starting in row F, highest concentration, diluting backwards up to the lowest concentration up at the top. And from there, again, on the spectrophotometer, and we can generate IC50 curves to read off the IC50 for each one. Okay, so literally the same principle as the parasite assay. Okay, so why do we do that? Well, what is the whole point of selectivity? What are we trying to do here? So the big important question you're trying to ask yourself is, do I have a good compound or do I just have a toxic compound? You can get a lot of things that will kill the malaria parasite. Bleach will kill the malaria parasite. But of course, if you get sick with malaria, well, look, I mean, you could drink bleach if you wanted to, and I promise you at that stage, malaria will be the least of your worries. So what we're trying to find out all the time is, do we have a good new compound, or have we just produced more bleach? So this is why we would run cytotox. It's a sort of a useful and at the same time sort of a useless assay, simply because it doesn't tell you very much, right? We generate what we call a selectivity index, which is the ratio of the cytotox IC50 to the parasite IC50. If you want to move your compounds forward, you need an SI of at least 10, but this doesn't tell you very much. So the hassle we've got here is, depending on your system, you may run it in HepG2 cells, which are human liver cells. You may run it in CHO cells, which are Chinese hamster cells. You may run it in Vero cells, which are kidney cells from the green monkey. You may run it in L6 cells, which are thigh muscle cells from a rat. And those are not the same type of cell. So if you get cytotox in a CHO cell, does that mean you've got a hideously toxic compound, or does that mean you've produced an antibiotic for CHO cells? So it's a weird result to look at when you get a selectivity index or a cytotox IC50. It gives you just a very general idea as to whether or not you've got a parasite-specific compound or something which may have general widespread toxicity. So believe it or not, this is not the be-all and end-all result that you get. We need it in terms of being able to do other work. If you want to do any sort of animal work at all, the ethics committee will want a safety margin of some sort, so they'll need to know what that SI is, but it doesn't always tell you anything. And if you look down at the bottom there, so we said, right, an SI of greater than 10 is usually required for any sort of animal work. Here are a whole range of parasite and tox IC50s to calculate the SIs, and you can see they're all greater than 10, but they are not the same value, right? So in the case of the first one, we've got a parasite IC50 of 8 nanomolar, really, really good, but our tox IC50 is only 100 nanomolar. So although you've got that 10-fold margin, you're still looking at something which is really, really, really cytotoxic. In this case of the second one, we've now got a significantly better tox IC50 and a slightly worse parasite IC50. You still have a good selectivity index of greater than 10. But in this case, well, this activity is probably no good to you. Bearing in mind, something like artesunate, which is the current gold standard antimalarial, has an IC50 of 2 nanomolar. If you've got a compound with 800 nanomolar, no one is going to ask you to please make that into a drug for widespread use. You're going to have a pill about this size. It's something you could kill a horse with, simply because the amount of drug needed to get to 820 nanomolar in the blood. So although that SI is good, this value is not necessarily useful to you. Okay, now what if we've got, say, maybe, significantly better activity and significantly worse tox? We've now got a much better selectivity margin. Is it good? It's okay. Again, believe it or not, right? So artesunate is 2 nanomolar. So 80 nanomolar is better than 800 nanomolar, but, well, it's still not quite potent enough. We'd be looking for something probably with an IC50 at the end of the day of less than 20 nanomolar is what you would want your final compound to be. Finally, we come down to the last one. Again, well, our SI now is 931. It's good, right? This is good. This compound is pretty selective. But actually, when you get further down your drug discovery process and you go from formal hit assessment to lead optimization to early lead to late lead, these numbers will change. By the time you get to your lead, SI doesn't have to be greater than 10. It has to be greater than 1,000 in at least 2 different cell lines. So although we do this, we do cytotox, we try and give you an idea of how good your compound is. It's a very, very preliminary result. You've got to view it in context. It's not something that you can just sort of say, oh, my SI is 10, cytotox is 100 times better than the parasite, don't worry about it. It's something you've got to take into consideration with all your other results put together. Okay. At that point, you ask yourself, if we've got a good compound, when does it become a good drug? And that is a whole nother story, right? You only become a good drug when you have good drug-like properties, and that's something that your DMPK department determines for you. It's entirely out of our remit. Cool. How are we doing for time? There's a lot of slides here. I don't know how much time you want to spend on this. We may want to at some point perhaps have a second one. We're only about halfway through, and I don't know how busy you guys are.
Okay, so what slide number are you on, sir?
Number 20 out of 42.
We are nowhere.
It'll go a bit faster from here, but we now move away from the standard assays into the little bit more advanced assays. So this might be a good spot to break, or we can keep going if you guys. I'm easy for time. It's entirely up to you.
Yeah, so I think we can break, and then we can have a more interactive session another time when you're free again.
Cool. Let me know when it's good for you, and we'll make it work.
Okay, Dale. Yeah, I think we can. I mean, if this is a good place to stop for you, we can stop and reschedule another one. It's quite unfortunate because this one we tried recording, but